Welcome to the Royal Society of Chemistry Atomic Spectroscopy Group's introductory video on sample analysis. The aim of this two-part video is to provide a step-by-step guide to the analysis of common materials. Part 1. Soils. The first step is to select a suitable sampling site. This may require cleaning the surface debris first. Samples can be collected in suitable containers such as soil bags, which can be closed and readily dried in situ. Containers should be clearly labelled in pencil. This process is repeated at all sites. It may be necessary to take samples at depth, for example, with an auger. Again, the sample is collected in a suitable container, here into a soil bag which is flexible. The next step is to dry the sample in a suitable oven, in order to prepare it for the next stage. The temperature is chosen, usually between 40 and 105 degrees centigrade, to avoid loss of analytes of interest, while removing the variable water content. Samples are placed in the oven for periods up to 48 hours, dependent on the temperature and quantity of material. When dry, the samples are removed and stored in a desiccator until required. To allow a more representative subsample to be taken, the dry sample is ground to reduce the particle size and then sieved to retain the smaller fraction. A typical particle size collected would be below 0.18 mm in diameter. The fine fraction collected is stored in a clean, dry sample bottle, which is clearly labelled. Before weighing the subsample, the analytical balance must be checked to ensure the calibration is correct, using certified weights of known mass. Here a 10-gram weight is being used. Next, the subsample is weighed into a suitable pre-cleaned dry container. A laboratory beaker is being used with approximately 0.25 grams of subsample. The exact weight is accurately recorded. In order to get the analyte of interest into solution for later measurement, we need to select a suitable digestion method. For our sample, the acid, in this case aqua regia, a mixture of concentrated hydrochloric and nitric acids, is added to the samples. Great care should be taken during this procedure. Note the use of gloves and a fume cupboard. One of the more common digestion methods is the hot plate open vessel approach. The samples are placed on the hot plate and covered with a watch glass to reduce the rate of evaporation. Note 3 replicates of each sample for each site. A procedural blank and a certified reference material are being used here. This digestion process often takes up to 2 hours and at a temperature just above the boiling point of the acids. Once cooled, the digested samples are diluted with ultra-pure water, then filtered into a volumetric flask, washing out the beaker's contents to remove all of the sample and rinsing with water or dilute acid. This is known as a quantitative transfer. This is repeated for all the sample digests. At the end of this process, the sample solutions are made up to the known volume and mixed by inversion. Before we can prepare our calibration standards, we need to calibrate the pipettes. This is performed by using the weight of the water they dispense. We can now prepare our calibration standards using, in this case, single calibrant solutions of known concentration of the analytes of interest. Before making up to their marked volume, the acids used in the digestion are also added to match their concentration found in the sample digests. The flasks are then made up to the mark and mixed by inversion. Finally, the measurement step is performed. Here we're using an inductively coupled plasma optical emission spectrometer. To construct a calibration graph, that is the response of the instrument to the known standards, each of the calibrants are introduced and measured in ascending concentration. This instrumental technique is related to the emission-based flame tests you have probably performed in your studies, but here the intensity of emission is measured quantitatively using an alternative hot source. A straight-line calibration graph should be produced, and this is used to calculate the concentration of the analytes of interest from the sample's measured response.